So first of all, I want to thank you for inviting me to talk to you. It's a pleasure to share my enthusiasm for trilobites with all of you. Um, I began in paleontology over 2 decades ago when I began my undergraduate degree. I came to the University of Iowa thinking I wanted to study vertebrates or dinosaurs, and the first project that opened up to me was working with trilobites. And I fell in love with arthropods through that and haven't looked back. So, my goal with this presentation tonight is to answer a question that, well, frankly, nobody's asked tonight yet. And that question is, why should we care about dead things? Things that have been dead for millions and millions of years. What could we possibly learn from these long dead creatures? And what I aim to do with this talk is answer the question that I myself have asked and show you the world of things that we have learned from these long dead creatures. So, first of all, what is a trilobite? I have a feeling most of you already know this. They are a group of marine arthropods distantly related to horseshoe crabs. Those of you can still see my picture there. I've got my nice little trilobite toy here. Trilobites were around for almost 300 million years. So they beat the dinosaurs in longevity. They lived from the Cambrian until the Permian. What makes them such iconic fossils is this. They have had a mineralized dorsal exoskeleton, a shell, if you will. And because it was mineralized, that means that it stood a much better chance of getting into the fossil record. So not only do they have a good chance of making into the fossil record, but they're also arthropods. So in order to grow, they had to molt, which means that they make lots of copies of themselves as they grow up that we can find in the fossil record. Well, this is just another picture of the trilobite body. Underneath that dorsal exoskeleton, that shell on the top, there was an unmineralized part that had the antennae and the legs. That body cuticle was not mineralized, and so it's much less common for us to find that in the fossil record. Now, trilobites have been called beetles of the sea, and that is just in reference to the absurd diversity of beetles that we have alive today, and comparing that to the absurd diversity of trilobites that were alive in the Paleozoic. I've got several examples right there of various types of trilobites. These are mostly, well, they do span the Cambrian until the Devonian here. So there's a good sampling of spiny, weird beasts. Now, I want you to look at that trilobite there that I have in the lower right or lower left corner. And think about that. I have a feeling many of you in the audience have collected fossils before, and usually that takes the form of taking a hammer, hitting something until it splits. That trilobite right there was not a product of that split. That was prepared out using air abrasives and little handheld jackhammers. And so I just want you to marvel at the time, effort, and skill required to release that trilobite from the rock. Now, this picture right here is sort of the state-of-the-art understanding of how the different groups of trilobites are related to one another. And what this represents, I want you to show you that the lineages all kind of go back to one central point. Just a second here, I have a minor emergency to deal with. Thought I had a bit. Someone needed a Band-Aid. I apologize. So all of these lineages are coming back to one point. That means that that's a sign of ignorance in a phylogeny. Since we don't know which groups are more closely related to what, we just pull them all down to the same point. This is sometimes pejoratively called a bush phylogeny because everything is originating from one little point. It's not a tree, it's a bush. The phylogenetic analyses that have been done of trilobites typically have focused on little branches. And this radiation of them that occurred in the Cambrian and Ordovician is still shamefully poorly understood. Now, if we just start counting up the number of different types of trilobites that we have, this is a diversity diagram, Cambrian at the bottom. It gets younger as we go up, and the bars represent the number of families. And you can see the diversity of trilobites is highest in the late Cambrian and through the Ordovician. We can see the effects of the Ordovician extinction here pretty easily, where the diversity of, or the number of trilobite families basically gets cut in half. It goes from 42 to 19. The end Devonian extinction is not as obvious on this diagram of trilobites, but it represents a major winnowing of morphological diversity. After that, trilobites all look the same. Now, the Cambrian and the Ordovician, trilobites were much more adventurous with their body shapes and sizes. After the Ordovician, they become a lot more stereotyped into lineages that we understand a lot more. And then I'll just point out lastly that they survived right up until the end of the Permian. At the end Permian mass extinction, they went extinct, and they were in good company because a lot of things went extinct at the end of the Permian. So back to the question that I posed at the beginning of this lecture. What do long dead things have? What could they possibly teach us? Well, I've organized this into 4 different stories here that they can help us learn about diversity. They can help us learn how we grow. And then they can help us learn about what I call winning the fossilization lottery. And lastly, maybe a bit of a stretch here, they can help us learn about technology, right? So let's begin. Part the first, trilobites and diversity. So this diagram right here is a retelling of the family diversity diagram I showed you in the previous slide. And not only can you see this big increase in diversity here in the Cambrian, but also you can see, this big increase or decrease at the end of the Ordovician. It is the first of the big 5 major mass extinctions. And this is showing that end Devonian decline much better than the other diagram. I think in part because this is a more recent diagram. So we go from high diversity to fairly low diversity after the end Ordovician extinction. All right. So remember that high in the Cambrian, the Ordovician. Post Ordovician, so Silurian, Devonian, low levels. All right. So there's our the Cambrian Ordovician boundary, and there is our extinction event. Now, if you've ever collected trilobites, there's a pretty obvious pattern that you begin to see. If you go to Cambrian age rocks, right? This is an example of a bedding plane of Cambrian age rocks from China. Every single fossil that you see in that picture is a trilobite. In the Cambrian, trilobites are the fossil, essentially. If you find other things, you are exceedingly lucky. Trilobites, you can't help but find them and see them everywhere. Now for comparison here, this is an Ordovician bedding plane I found online, illustrated from Minnesota, presumably from southeastern Minnesota. And right away, I want you to notice that there are lots of different things here. There are trilobites here, but you've got to look hard to see them. And so think about this with our trilobite diversity diagram. I think given the Cambrian picture where you just see trilobites, that makes sense. High diversity, yeah, they're the only things there. In the Ordovician, things begin to change. Now, I began doing fieldwork for trilobites as an undergraduate, and I think a lot of people envision paleontological fieldwork, something like that picture that you see there. That's not what I do. That is a much younger and slimmer me. What we would do is get our backpack and a sledgehammer. We would hike up the mountain, use the sledgehammer to fill up the backpack, stagger down the mountain, empty it into the truck, and then just repeat until it got dark or we got hungry or exhausted. We weren't doing careful sampling. And the reason for that, and this is another picture of myself and a colleague in Nevada at Patterson Pass, is these rocks were special. We wanted to send them all back to the lab without doing any selection at the site. And so we would put them in 5-gallon buckets, take them to UPS, and then we'd have the same joke every time we'd go to UPS. We'd lift the buckets up onto the counter, and the clerk would say something like, "What do you got in here, rocks?" Yes. Now, I mentioned that these rocks were special. In these rocks, the normally calcium carbonate, calcite skeleton of the trilobite was replaced with silica, same stuff that's used to make glass. What that means is that we can take that limestone, put it into hydrochloric acid. The acid will eat away the limestone and leave behind piles of fossils. And I've actually got some here. This is a fossil residue right here, the fossil rich stuff at the bottom of the bin when we're done, of the bucket when we're done dissolving it. And then you put that under a microscope and you can pick out all the trilobits that you can find there. This one has a lot of other things in it as well, in addition to the trilobites. Now, because they're replaced with glass effectively, they're very fragile. You will break them, and it's just something you have to get used to. The detail of these kind of specimens is astounding. When you have a crack out specimen, it's on the surface of the rock. You can't change that. With these silicified specimens, you can see every side of it, top, bottom, front, back. So it's three-dimensional preservation. And as a result, it is excellent for understanding and observing the morphology. There are lots of features of trilobites that we just could not see in crack out material. We can only observe them in this three-dimensionally preserved silicified material. Now, the old timers would just go out in the field and grab a block, a couple of kilograms, take it back, dissolve it in acid, and that was the diversity that they found. Work by my former advisor, Jonathan Drain at the University of Iowa, suggests that you probably need about 50 kilograms of rock dissolved in acid in order to get an accurate picture of the diversity. That's 50 kilograms to find all of the less common and rare things. Now, this gives us a new way to think about diversity, okay? We looked at, earlier in this lecture, the sort of global diversity of families. Well, what my former advisor did is he looked at how many trilobite species you find in each particular habitat. all right? So each paleo environment. So we have Laurentia right there, which is the paleo continent that today we call North America. And if we look at near shore habitats, in the Cambrian, that's the sea there, we find about 4 species. In the Ordovician, about 3. In the early part of the Silurian, called the Llandovery, we find about 5. Later in the Silurian, in the Ludlow, we find about 4. So basically, through the Cambrian, through the Silurian, we get the same number of trilobite species in near-shore habitats, okay? And as you look there, that pattern essentially holds everywhere where we've got significant data, with a couple of weird blips like the diversity of deep subtidal habitats in the Wenlockian part of the Silurian, for example. But essentially, through Cambrian, through the Silurian, you have the same number of species in each habitat. All right, now let's put that in context. Remember that diagram? We had a huge drop in the number of species globally, or number of families globally. We don't see that in the individual habitats. Why? What's going on here? Well, this is a kind of a strange phenomenon, but let me give you an added detail here. If we go to the Ordovician and we look at those 3 species that are in nearshore habitats, they will be completely different from the 12 that you find in shallow subtidal, and those 15 will be completely different from the 10 you see in carbonate buildups, and those 25 will be completely different from the 23 that you see in the deep subtidal, and all of those completely different from the 4 in the basinal deposits. All right, so that gives you a total diversity of about 50. If we move into the Silurian, what we see is that instead of each habitat having a completely different set of species, what we have is the same cast of characters, but in each habitat, they sort of change in relative abundance. So those 5 nearshore species, you'll see them still in the shallow subtidal. It's just they're going to be less abundant than they were nearshore. And the 13 that we're seeing in the carbonate buildups, well, that includes most of this, that includes the 11 from the shallow subtidal and some of those nearshore ones too. So the net diversity across the Llandovery there at the early Silurian would only be 13. It's not additive because it's the same critters in each habitat. It's a phenomenon of environmental differentiation, something called beta diversity. Beta diversity between habitat diversity crashes at the end of the Ordovician. Now, what we would like to think when we take those blocks of limestone back to the lab and dissolve them is that that is a complete record of all the trilobites that lived in that environment, right? Everything that lived and died left their bodies right there for us to find. If that is true, we should always find a 1-to-1 ratio of heads to tails. You look at a trilobite, like my toy here, it has 1 head, 1 tail. Even though it molts and it grows a new exoskeleton, throws that off, the old one off into the fossil record, that old copy still has just 1 head, 1 tail. Okay? So you should always have that ratio. If you don't, that means that something else has happened, like currents or waves washing away different sized pieces. And so my former advisor came up with a really neat way to test for this phenomenon. He noticed that some trilobites from the Ordovician of Utah, like the A, B, E, and F there, are what we call micropygous. That means They have big heads and little tails, relatively speaking. And then there are others like C, D, G, and H, which are what we call isopygous. That means the head and the tail are about the same size. So if there's a different behavior of these trilobites due to the currents in the water, we would expect the big pieces to behave differently from the little pieces, and thus we'd get a skewed ratio. And so that bar chart there at the top is showing the results of that. Instead of always getting a 1-to-1 ratio of heads to tails, that ratio gets as screwy as 200-to-1. That means that those smaller, lighter tails of the micropygous species are getting washed out by currents that we didn't think were affecting these habitats. That means that there is a profound and pernicious bias in these habitats, that what we find is not necessarily a true record of the diversity there. It's already been filtered by these currents. Now, that phenomenon of silicification, that magic replacement that allows us to study these fossils, itself has not been well understood. And we know the fossils started out as calcium carbonate, as calcite, and something happens, and then they're replaced with silica. Replacement is a pretty normal mode of fossilization. It happens with pyrite, it happens with calcium phosphate. The problem is we can get pyrite replacement to happen in the lab. We can get calcium phosphate replacement to happen in the lab. Silicification, really, we've not been able to duplicate in the same way in the lab. So it's been frustrating. This picture here is kind of a nice one because it shows what these silicified fossils look like when you're out in the field. Because the weakly acidic rainwater eats away that limestone and leaves these silicified fossils really sticking out on the surface. Now, for trilobite workers like myself, we kind of wanted to believe that that silicification was a perfect 1-to-1 replacement. And as a result, that the fossils that we got out were a perfect silica replicate of the morphology of the trilobite. No weird little changes that the silicification made. However, there's sort of a fundamental puzzle to this that maybe we didn't give enough attention to. First of all, since the surrounding rock was calcite and the trilobite was calcite, you have to ask the question, how did this process know where the boundary of the trilobite was? If it was just replacing calcite, shouldn't it have just gone into the calcite mud after it was done with the trilobite? Second, the replacement process itself. You can't have two minerals occupying the same space. What's going on? Do you just have the calcite go away and that space just stay there for the silica to come in? Seems kind of unlikely as well. So into this puzzle, I want to show you the sort of spectacularly innovative research that can happen when I go away. And so when I left WIU to go to SUNY Fredonia, I had a research student working with me. And I was kind of abandoning him, unfortunately. So I left him a pile of silicified material, kind of set him up with a project where he would image the material on the SEM, scanning electron microscope, and then he'd try to do a phylogenetic analysis of the group. Great. Well, I had to get somebody to supervise him on the expensive scanning electron microscope. And so my colleague graciously agreed to do this. Her experience, however, was on microbially mediated cave structures, how microbes help form the calcite in cave pearls and flowstone. And so as a result, she was used to using the SEM, but at a very, very different scale. And so what they began to do is he would get the SEM focused on a specimen, and there'd be something in that field of view that would trigger a memory of and cause her to say, you know what? Why don't you zoom in right there? Zoom in more. Zoom in more to a degree that would be ridiculous for any trilobite worker. But this texture is what showed up. And I want you to notice there that you have places where there are these nice crystal boundaries, I think molds of calcite or dolomite probably. You get this sort of kind of slimy looking areas where things are kind of, it's like there's a film draped over it. And you've got other places where there seem to be maybe clay grains from the matrix actually stuck to it. And when you keep looking closer, you find even more interesting things. Notice in this SEM image here, these ropey things that are on this kind of draped matte surface. Those ropey things we think are microbial filaments that have been replaced with silica, and that draped structure there, smooth, slimy look, that is the silicified extrapolymeric substance of the microbes, the sort of slime that microbes make. This is really weird, right? We thought these were a one-to-one replacement, but instead we have fossil microbes from the Cambrian here. And what we think is that these were the missing ingredient in silicification, that the reason we weren't able to get silicification to work in the lab is because we didn't have all of the ingredients. We were missing the critical biological ingredient the whole time, and we didn't realize it. Now, if we're right, that predicts that we should be able to look at silicified material from anywhere on the planet, any age, and we'll see these same sort of patterns. And so that's what we've been doing over the last year. And it seems like so far these patterns are universal. I do want to point out here, you could rightfully argue that maybe these are contaminants of some sort. Well, if they are, when we do an elemental analysis with an EDS spectroscopy unit on those filaments, they should show up as carbon, not as silica. And you can see right here that we get that strong silica peak, as well as a strong silica peak there on the filaments as well. We're getting some carbon, but it's got a strong silica peak as well, which is telling us that there's replacement going on, that this is not a modern contaminant. Now, in the process of looking for this pattern elsewhere, we've stumbled upon something else quite unexpected. We had samples of some Permian brachiopods, silicified, from the Glass Mountains in Texas. And when we began looking at the internal surface of them, we began seeing these weird pill shapes. Those pill shapes, when you do a spot analysis on them, they are made of iron, they are made of sulfur, they are pyrite. And the weird thing is they look kind of like a pyrite structure called a framboid. Framboid is French for raspberry, and you can see one there in B. It's that big circular structure. Pyrite, sometimes in mud and anoxic sediments, will just grow into these little round balls. They're always tiny. These pill shapes, right? They're the shape of bacteria, but they have the framboid style filling. And what we think is going on is that you've got this framboid style growth, but it's happening within the confining membranes of a microbe of some sort, bacterial cell. Now, we thought we were onto something really new, and there's just been a preprint in the last 2 weeks that has come out showing these same kind of structures from the Cambrian, not from the Permian like we've found. So somebody beat us to the punch, but I think that's going to actually help us understand this a little bit more with this second data point. Now, trilobites and growth. The fossil record of trilobites, the fact that they molt and throw a copy of themselves into the fossil record as they grow, means we have a very good opportunity to study how they grow. And so there have been a couple of iconic cases where people have been able to assemble this growth trajectory to understand how trilobites go from a little shield-shaped baby the size of a period at the end of a sentence to an adult. And so this right here is one of the best examples of a trilobite growth pattern that happened. Yes. This is one of the best examples that we have. It was put together in the early part of the last century. This is a trilobite called Shumardia. It starts out as that little round featureless shield. As it grows, it divides that shield into a head and tail, and then begins to add segments in between. But here's the thing, it's not actually adding segments in between. Segments are growing right at the back, and they are conveyor belted forward as new segments form behind them. And you can track that in this trilobite here, because the adult has this structure here that we call a macropleural segment. It's a big segment, different from all the others. It's always fourth from the head. But if we watch it grow, right, it originates as points at the back of the tail, and then those points move forward, so they're not at the back anymore. And then they move to the front of the tail, and then it gets released as a segment, and then another segment gets released behind it, and a second segment released behind it. Segments are being conveyor belted forward. That's how these arthropods grow. And in fact, that is a more general phenomenon in how arthropods grow. Many arthropods grow. They have a growth zone at the back, and they kind of generate the segments there and move them forward. This is something I've done some work on, doing developmental work with some modern branchiopod crustaceans like fairy shrimp and tadpole shrimp. But we're seeing that same pattern way back in the Paleozoic. Now, we typically think of animals as having an adult shape, right? You go from baby to adult. And in trilobites, that would mean getting the adult number of segments and then growing bigger after that, right? Well, one of my colleagues, Nigel Hughes at UC Riverside, stumbled upon an interesting pattern with this trilobite here called Aulicopleura. Aulicopleura konickiae is a absurdly abundant little trilobite in the Czech Republic. And in the places where you find it, you can find everything from the babies to the adults. And so what he did is he started counting the number of segments and comparing that to the length of that little round spot in the middle of the head called the glabella, right? That glabellar size is meant to kind of show us overall growth. Now, when we look at other species of trilobites, so that's all of these other shapes here, notice that they get to a number of segments, like these triangles here, And then they just grow larger. They don't add any more segments. Every other trilobite is like that. They grow up, get bigger, grow up, get bigger. This smearing up here of Alocapleuroconicea means that some of the big trilobites, the largest ones, had as many as 22 segments. Some of the largest ones only had 18. So why is there this variability in the number of segments? It's not even sort of a sort of dimorphic variability that we say, oh, it's two groups, so it must be males and females. No, it's not like that at all. What's going on? Well, one of the ideas for this is the deposits where these are found seem to be low diversity, low oxygen environments. Basically, not much oxygen, so very few creatures could live there. And since trilobites have a pair of legs for each segment, and on each pair of legs they have a pair of gills, adding more segments, having that flexibility would be a way to add extra oxygen breathing ability, right? An extra set of gills to pull oxygen out of the water. So it may be related to this low oxygen lifestyle. Whatever it is, Alocapleuroconicea seems to have broken one of the rules of how trilobites grow. That there's not a fixed adult shape or size, I guess. Now, trilobites have become classic examples in thinking about how big changes happen in evolution. We think of these big changes sometimes as being like a feature just pops into existence. Well, that maybe can happen. The problem with that is that all of the other members of the species need to still be able to recognize that weirdo with the new feature as someone of the same species, someone they could mate with. People have talked about those big sudden changes as creating hopeful monsters. And to that I say the hopeful monster is probably the sexually frustrated monster, because nobody's going to want to reproduce with them. They don't look right. Well, one of the ways that we can get big changes to happen in evolution is by changing developmental timing. This is heterochrony. That name, heterochrony, literally means different time. And so in this diagram right here, at the bottom, we have an orctocephalid species of trilobite. The one is showing us the baby. The babies are relatively smooth, have a head and tail that are about the same size. And as it grows up, see 6 and 9, they get increasingly smaller tails proportionally and a bumpier head. Well, if we look in this formation in China where we get later representatives higher up in the stratigraphy, younger, see that they get increasingly smoother, and they get increasingly proportionally larger tails. All right. Basically, what they're doing is they're keeping those features that were baby features in the ancestor, but they're now keeping them as adults and becoming sexually mature sort of earlier in the growth cycle. One of the great examples of this sort of pattern is the axolotl, the salamander that never grows up. It keeps its gills. It never metamorphoses into an air breather. It stays in the water. It just moves the timing of sexual maturity earlier in its growth pattern. Here's another good example of this same kind of pattern. On the left, we have Olenellus lapworthi. The adult is at the bottom. Above it, though, is what the baby looks like. The baby, instead of being that smooth shield that we saw before, is an incredibly spiny shield. It was kind of like an asteroid. And what we see in these related species as we get younger and younger in time is that spininess of the baby is kept. These spines going backwards from the head, the genal spines, kind of migrate around the head. And that's sort of reenacting or undoing the process of growth that the ancestor did. Trilobites and fossilization, part the third. So trilobites, as I said before, have a mineralized top and unmineralized undercarriage. And usually it's only that mineralized part that survives. Got a good example of that here that I can show off. This is a trilobite cluster from the Ordovician of Oklahoma. And here we have trilobite bits and pieces all over the place, but no soft parts. Now, if we get soft parts, that means something very different has interrupted the fossilization process. And usually that is low oxygen conditions, because those are what keep predators, scavengers, and microbes mostly away. If you can't breathe, you can't eat. And there's also usually changes in geochemistry accompanying them. So in the sort of normal deposits, we'll find maybe the exoskeletons, the results of generations upon generations of trilobites molting, and then going off and doing their thing, leaving copies of themselves in the fossil record. But sometimes those hard parts are weird. So at the top of this image here on the right, we have some trilobites with spots. These are from the Devonian of New York. And I and some colleagues published on these about a decade ago. And we looked at those spots. We sliced up some of these nice trilobites to look at those spots under the scanning electron microscope. And what we noticed is those spots kind of sort of looked like the lenses in the eyes. And so one of the things that we thought is that maybe these are not eyes per se, but sort of a window through the exoskeleton for something like a chromatophore, which is a color changing organ in like squid. So maybe these were sort of a camouflage adaptation. At that time, we had only seen these type of spots in this one species of trilobite from this one place at this one time in Earth's history, never anywhere else. Well, just this last year, I've realized that we were wrong, and I'll show you why. Here is a specimen. Let's see if I can get it to show here. We've got this very spotty tail there. That's from this species right here. Notice that the tail, it has a smooth boundary. We've now found a second species with spots, still the same time period, still the same place, but now we've got a completely different family of trilobites, or subfamily, with spots. Notice the spiky tail. What's going on? Before, we were thinking of this as sort of a unique feature developed by one particular species. Now, I think the odds of two different species in two different families figuring out the same trick is kind of low. And what a coincidence that two species that figured out the same trick happened to do it simultaneously, but only in this one particular place at this one time. It kind of stretches belief. So we're beginning to explore some other interpretations of these spots. So stay tuned. One of the ways I like thinking about these, though, that I think is really interesting, is to think about them as if they were World War II bombers. I think probably a lot of you have seen a picture going around on the internet of a World War II bomber, and mapped onto this outline of a plane are bullet holes. What they were doing is tracking where they found bullet holes on the planes that came back. And sort of the naive response to that is to say, oh, well, if they get shot there, we should put more armor there. What you're not considering, though, something called the survivorship bias. Your sample set is only the planes that were able to fly back. In other words, those places that you documented bullet holes, those are the places where it is survivable to get shot. The places where you never see bullet holes, those are the places that cause you to crash and never come back. And I wonder if the patterns on these trilobites maybe are a repair phenomenon, and that we see them localized only because these are the spots where they could survive and repair them, and they weren't fatal. Now, the trilobites here at the bottom, those are the center part of the head called the glabella. I want you to notice those dark spots. These are kind of superficially like what we see there, except these have been noted in lots of different trilobites all over. What they seem to be is the segmented muscle attachment sites for the legs in the head. The puzzle that we've been trying to work on with them, though, is why are they dark? And really, that gets at a more fundamental question. Remember, trilobites molt. They take off a copy of themselves when they grow, but the muscles are attached to the skeleton. How does that happen? And we've got good ideas of how that happens in modern arthropods just from watching the process, documenting it, studying it. But trilobites, that's a little less clear. Now, pictured here to the right are a couple of spectacularly preserved trilobites from the Burgess Shale in Canada. They are actually the real fossils that this little model was based off of. And notice that instead of just having that hard exoskeleton here, you have legs, you have antennae. Now, this tells us that something different happened during fossilization. Something deviated from that normal pattern. As I like to put it, they won the fossilization lottery. And so if we want to interpret these structures here as part of the biological organism, we've got to figure out, how did they get into the fossil record? So a neat example of this that I was involved in involve some gold trilobites from upstate New York. And I want to show you an example of one of them right here, although the lighting in here probably means that you won't be able to see it that well. All right, this is a little trilobite called Triarthris eatoni. And these trilobites are made out of pyrite. They're made out of fool's gold. They've been replaced with pyrite. Not only the exoskeleton, but also the soft parts, the legs and the antennae. These replaced trilobites with legs have been known since the late 1800s, but our tools to reveal them were relatively coarse, meaning they'd take like a wire bristle brush the shale off. A friend of mine began working at this site over a decade ago now, probably close to 15 years, and his goal was in part to get these trilobites, prepare them, and sell them. And he developed a really innovative technique for preparing them. He used the hardness of the difference in hardness of the shale versus the mineralogical hardness of the pyrite, and found I think it was actually a micro ceramic particle that is harder than the shale but softer than the pyrite, so he could air blast it at the trilobite and preserve these fine golden details. And as he perfected this technique, the trilobites that he produced had a lot more detail preserved. And he began noticing in about half of the trilobites that he looked at, that he prepared, he would find a cluster of little round things on the sides of the head. And so that ratio of about half And the presence of little round things kind of made him wonder, are these maybe eggs? So he showed me these pictures, and we began thinking about, well, how could we test this? What sort of argument could we make to think about what these are? And so we did a lot of things, from looking at them under the scanning electron microscope, to exploring modern arthropod biology. To a mammal like me, the idea of carrying eggs in your head is bizarre. But that's not bizarre to arthropods. In fact, horseshoe crabs do something very similar. In a formative event for me was realizing I could learn a lot about horseshoe crab anatomy by watching YouTube videos of Asian street vendors preparing horseshoe crab egg omelets. And I'll show you a picture of that in a moment. You will have to do your own YouTube searching to see the whole graphic video. Now, because these are preserved in iron sulfide and pyrite, we could CT scan them. And so we could digitally dissect out the eggs. And we noticed that they are very uniform in size. They've got a unique style of preservation that matches the soft parts of the trilobite, but not the exoskeleton, and so on. Now, horseshoe crabs, in the front of their head, the females have what they call an ovarian network. All right, this is basically an area that forms the eggs and holds them. And then the eggs are eventually released through two discrete little pores at the back of the head. The male gametes are released the same way. And so if this is a model for trilobites, this answers the question that I think all of you have probably thought about trilobites, but been too scared to ask. You look at one of the Burgess Shale trilobites and you say, well, where's its penis? It didn't have one. This is the answer to why we never see trilobite genitals. They were just discrete little pores hidden behind the legs. And so here's a picture from one of these YouTube videos of a horseshoe crab egg omelet. You can see there, those little greenish circles are the eggs. And what the street vendors will often do is cut the horseshoe crab around the margin of the head, rip off the legs. That's usually still alive at this point, which is what makes these videos rather disturbing. They will scoop those eggs out of the head there into a pan, and then drizzle the blue blood of the horseshoe crab on top. Yes, they have blue blood. They use copper instead of iron to move oxygen around. Trilobite guts. This is some work I did with a friend of mine, Rudy LaRossi-Abril, who's currently at Harvard. These are trilobites from the Weeks Formation, the Cambrian of Utah, and they're kind of famous amongst collectors. It's common to see it fossil shows. Some of the trilobites from the Weeks Formation show something unique. They have these axial structures that are sitting right where the digestive tract would be. In fact, they're not just in the trilobites. As you see there in H, they're sometimes in these soft-bodied arthropods as well. Here's the thing. Those exoskeletons are not well preserved. You look at them, they look kind of shitty. And they also don't have legs or antennae. Instead, they have a very well-preserved mold of the gut. How does your outside look like crap? The leg's not preserved, but the innermost parts of you, the guts, are preserved. Kind of wild. Well, it turns out that there are some facts about arthropod biology that help us understand this. Number one, when they're preparing to molt, molting can be a very wasteful process, right? You're throwing a whole copy of yourself in the trash. Well, what a lot of modern arthropods do is they pull some of the mineral ions out of their exoskeleton before molting, which is, I think, why these exoskeletons here look kind of crappy. Where do they put those ions? Well, they often have little glands, outpocketings of their gut, called digestive caecae sometimes, where they put them. The gut itself would have microbes in it, and it also have food. Amongst the things that the food would be rich in, presumably, would be phosphate. So you're getting the calcium in there, you're getting the phosphate, getting the microbes. And now the last fun fact that you need to know about trilobite guts is, or arthropod guts in general, is that they are lined with the same material that makes the exoskeleton. They don't put minerals in it, but it's lined with chitin, just like a bug exoskeleton today. So you have a chemically robust chamber in their gut, sealed at either end by a sphincter. You load it with chemical food, with mineral ions, and with some microbes. When the trilobite dies, those microbes don't just say, you know what, we've had a good run, let's just die with it. No, they stay alive and they stay hungry and they keep eating until they run out of food or some other limiting resource. And so that closed chemical chamber of the gut becomes sort of a chemical reaction chamber. And the activity of the microbes digesting the food that's left creates an environment where calcium phosphate now can precipitate, probably related to pH. So here, understanding the biology of the relatives helps us put together a scenario that helps us understand how this can happen. And I have sitting next to me here one relatively poorly preserved specimen from the Weeks Formation with just a little bit of the digestive tract preserved there, if you can see it. And that's not the only place that you see this. This is an example that was just published earlier this fall. Also, it's from the Czech Republic, I believe Ordovician in age. And what they had is they had a nodule that when split had a trilobite inside. But the trilobite exoskeleton was completely gone. They just had an exoskeleton shaped hole where that was. And so what they did is they CT scanned the nodule and then dissected out that hollow space inside of it where the exoskeleton was. And they found something remarkable. There were bits, shelly bits, still in the region of the gut. And they were identifiable. There were echinoderm bits. There were ostracod bits in the gut here. So this is spectacular evidence that this trilobite here was actually feeding on some mineralized things too and ingesting them. Plus, I am just absolutely in love with that 3D model. It's gorgeous. Trilobites and technology. I've got a couple of little stories to share. Two of them, frankly, if I'm honest, are kind of stretching the connection to technology, but the third one, that'll help. Now, there are a weird group of trilobites called the acidids. This includes a common trilobite throughout the Midwest, Ohio, Missouri, I believe even Wisconsin and Minnesota have some of them, called Isotelus. They have these bizarre plates underneath their heads. This is a mold of one of them. And these acidic trilobites got quite big. They're the biggest trilobites that ever lived, about 1 meter long. Why would they have these little forks on either side of their mouths? What possible purpose could they serve? Well, previous scientists had observed or had speculated that they were sharp and they were cutting edges. To me, that interpretation doesn't hold water. And here's why. First of all, when you look at them in 3D, they're actually kind of triangular in outline. I don't know about you, but I don't use triangular knives at home. They're flat, they're honed to an edge, sharp edge. Second, this plate would have been rigidly fused to the head. And so that would be like trying to eat a steak and cut it with your head, right? That's ridiculous. This is not something that would work. Now, because of this supposed sharp cutting surface, aciphid trilobites have been envisioned as big predators. And they are also large, so it was easy to imagine them sitting at the top of the food chain. Well, I began looking at these structures in a bit more closely in graduate school. And what I noticed is that inner surface there that faces where the mouth would be, there are actually little parallel lines there. They're actually terraced when you look at them under the scanning electron microscope. It's a washboard. It's a grinding surface there. When you cut one of these tines of the fork up, as you see in G there, that surface with those lines is as much as 7 times as thick as the exoskeleton anywhere else. So that's telling you that for whatever reason, this grooved surface has to be more robust than everything else. Those terraced lines also have that steep side facing the mouth, which would catch food as you ground it against this grinding surface. But I think there's maybe a more important thing to note here is that the nature of this surface close to the mouth suggests it was used for feeding, right? You don't grind up big prey. Lions don't grind up gazelles. Furthermore, if you have this over your mouth, right, eating a big struggling prey item, it's like trying to eat a sandwich through a football helmet. It's not going to work. So I think what these guys were doing was maybe like whale sharks or blue whales. They were giants, but they were feeding low on the food chain, eating something that didn't resist, that they could just grind up, process before ingesting. Now, when I wrote this paper in 2010, there was kind of a problem. And that is every trilobite that we knew of had legs coming out. They have antennae, and then they have walking legs. And the position of these walking legs doesn't really make sense with a grinding surface. So if I was right about this interpretation, that implies that these aciphid trilobites were doing something different with their legs. Otherwise, they couldn't use what I had hypothesized was a grinding surface. Well, it turns out we think we now have some evidence of this. And I'll be talking about this at the North American Paleontological Convention meeting this summer in June in Ann Arbor. The specimen there on the right is a specimen of Isotylus that was collected in the late 1800s. It was published by Elkanah Billings, a notable Canadian paleontologist. And what has happened is the legs are preserved in this as sort of kind of semi-molds. And there are some of these segmented bits that are really obvious and you can tell that they're legs. Others are maybe a bit less clear. But what is clear, if you look here closely, on either side of those forks are 2 little parallel ridges. I think these are legs that have been turned. And what's interesting is this seems to be a group of trilobites that's breaking a body patterning rule. The rule of trilobites is that you have antennae type limbs, and then you've got walking limbs, right? Those are your 2 choices. These aciphid trilobites seem to have maybe had another option. They specialized one of these pairs of walking legs specifically for feeding. And that happens in lots of modern arthropods, but it's always coupled with Hox gene shifts. Wow, I'm going long here. I apologize for this. I will try to wrap things up here quickly. Trilobites had calcite lenses in their eyes, which means we can study the mineralogical properties of them in a lot of detail. Broadly, there are these two types of eyes, the holochroal and the schizochroal eyes, which are different maybe due to looking stereoscopically, quite sure. Like I said, we can study the physics of how light goes through those lenses. And there's a new approach that we can now use called EBDS, electron backscatter diffraction spectroscopy, that maps the orientation of the crystals. I'm sorry, the janitor is coming by cleaning the floor here late. So this reds and oranges there in the picture mean that those crystal axes are all lined up. The trilobite very finely controlled the orientation of the crystal lattice specifically for its eye. Now, that kind of work intrigued me. So what I thought of were these weird aciphid trilobites that are found in the St. Petersburg area, Ordovician of Russia, that have stalked eyes to varying degrees. You've got a species there with low stalked eyes, you got the middle, and then the absurdly stalked eyes. And what always struck me as weird about this is those stalks are mineralized. They can't retract them. They're putting this valuable, vulnerable organ at the top of something they can't retract. Is there any adaptations that were different about that? Well, we began using electron backscatter diffraction to study this. And to begin with, everything looked kind of normal. All right, we can see the lenses there of the big stalk, lenses there, the medium stalk, and the small stalk. But then as one of the things you should know is that when you study trilobite eyes, you usually start with a really nicely preserved specimen, and then you're forced to slice it up. This is something that makes us tend to only use one representative because we don't want to slice up a bunch of nice trilobites. Well, with our multiple specimens, we realized that there were two different lens styles in two of the species. That Aciphus cornutus had both, some specimens had conical lenses, others had prism-shaped lenses. In Aciphus punctatus, there were ones with the conical lenses and ones with the parallel-sided prismatic lenses. Why this was, we don't know. We're still trying to figure that out. But lastly, the study of trilobite eyes has yielded a novel way to think about camera technology, a increased depth of field way to take pictures. All right, so I'm done. I hope I've demonstrated that there is a reason to study long dead things. I'd be happy to take any questions that anybody might have. I'm sorry for going late.
Joe, or Tom, excuse me. I was thinking with somebody else here. Got two questions up here posted. One, Marlene and Jim, would they bite you, and how fast do they travel? Do you know how fast these things actually moved?
No. I think probably crabs and horseshoe crabs are probably a decent analog here. There are some trilobites that we really strongly suspect were probably swimmers and not crawlers. But as far as speed goes, I think modern things are probably a good analog. Now, would they bite you? I think they might pinch you. So they might use the bases of their limbs, their shoulders, to pinch you, but they couldn't get their mouth at you. They would be frustrated trying to eat you.
Roger Benepi has asked, what molar concentration are you using to get the best results when you're talking about breaking down the limestones? Are you using a very acidic molar?
We're basically, so what we've learned is that you don't want to just put it in complete acid because that will yield to a really fast, violent, messy reaction. And so what we would do is we'd put, maybe 2 kilograms of rock into one of these buckets. We'd fill it up so water covered the block. And then we would add about, 500 milliliters of acid at a time. And then every day or so, we'd go in and have a block of limestone that we'd stick in to see if it reacted. Because if the acid was still there, that meant it was finished with what was in the bucket. If the rock that we stuck in didn't react, that meant it used up all the acid and we needed to put more in. I did, as an undergraduate and graduate student, probably some unsafe things as I turned our 500-gallon drum of, no, 50-gallon drum of hydrochloric acid into small portable containers.
Okay.
Always hairy, especially when I was trying to move that big drum by myself.
How about like that, like a 1 molar or a, do you remember the actual molarity?
I don't remember the molarity. It was 100% acid that we were putting in.
All right, real good. I'm looking here and I'm seeing lots of thank yous, and we have a lot of people who are listening tonight. We had over 50 participants, and so I'm going to let you get going. We're going to get going too. And thank you very much. And we will, just a second, I think got one more question that just came up, I think. Okay.
I see a question here. Yes, it is my daughter. And she hasn't learned how to prep trilobites yet, but she can polish rocks. She's good at that.
Mary Helen Linsky was asking, could a trilobite legs reach up to that washboard? Could it possibly shred food against it? Would that work, do you know, or any guesses on that?
Yes, but think about the orientation here. In most trilobites, they're oriented the wrong way to really rub against that surface. And so
Okay.
the trilobite that I showed, it looked like the limbs had been rotated about 90 degrees, which meant that they could oppose that surface effectively.
Well, great. Well, thank you very much. So let's get going, and we'll go to everybody. Have a good night.